Beta-thalassemia is one of the most common blood disorders in the world, particularly in Southeast Asia and North Africa. Research that appears in Science Translational Medicine details a new promising approach for this blood disorder. Beta-thalassemia is an hemoglobin disorder. Hemoglobin is a protein in red blood cells that carries oxygen from lungs to all tissues in the body. Hemoglobin is formed by 4 different globin subunits, 2 alpha globin subunits, 2 beta globin subunits. Beta-thalassemia patients inherit a point mutation that decrease beta globin synthesis. The biochemical hallmarks of this reduced beta globin synthesis is accumulation of toxic free alpha globin proteins. Free alpha globin subunit is toxic for the cells, will precipitate and will shorten half-life of red blood cells, but also destroys progenitors in a process termed ineffective erythropoiesis. Beta-thalassemia causes morbidity and early mortality of 100,000 people worldwide. So far, the only treatment for beta-thalassemia are blood transfusion and medication to limit the iron accumulation from those transfusions. But many of these patients do not get access to this treatment. We have identified an enzyme named ALK1, and this protein acts to increase the clearance of free alpha globin in these red blood cells through autophagy. Autophagy is a process from the cells to clear toxic protein. The loss of ALK1 in those mice from beta-thalassemia increases accumulation of alpha globin subunit and worsens the phenotype. Previous studies indicated that ALK1 could be inactivated by another protein named mTOR. Therefore, we hypothesized to use an mTOR inhibitor named rapamycin to improve ALK activity and increase clearance of free alpha globin in those beta-thalassemic mice. The electromicrograph shows reticulocytes from mice treated with rapamycin. As you can see, these drugs reduce significantly free alpha globin precipitate in those mice. More importantly, this effect of the drugs occurred only when the ALK1 gene is present. Rapamycin also reduced free alpha globin accumulation in erythroblast in culture from patients with beta-thalassemia. These patients are still under care in Milan, Italy. This project was a long-standing collaboration between 2 research labs at St. Jude Children's Research Hospital, Dr. Weiss and Dr. Kundu's labs, but also we got help from 2 expert clinicians from Italy, Dr. Capellini and Dr. Motta. In conclusion, we showed that the loss of ALK1 gene in thalassemic mice reduced the clearance of free alpha globin and worsened the phenotype. The rapamycin treatment improved ineffective erythropoiesis and red blood cell survival via an ALK1 dependent pathway. Rapamycin is FDA approved, inexpensive, and we can manage the toxicity of these drugs. Our next challenge will be to test a proof of principle clinical trial to see if these drugs could be effective and safe for patients with beta-thalassemia.